in the last activity, a sample of Sue's lymph fluid was sent off to the lab in order to identify the pathogen that's making her sick. The DNA will be isolated from her sample, and by determining the sequence of the pathogen's DNA, we'll be able to determine its identity. Before you complete this analysis, I want to give you some background information about how DNA sequencing works. Remember that all DNA is made up of nucleotides, A's and T's and G's and C's, and it's the order of these nucleotides that differs among different organisms. DNA sequencing is how scientists determine the precise order of nucleotides within a molecule of DNA. Many different methods are used to perform this process, and now next generation sequencing methods can sequence an entire human genome within a single day. For our purposes, we're going to focus on a common method used to sequence DNA for clinical use called cycle sequencing. First, the DNA is isolated from the sample. Once we have removed the DNA from the rest of the sample components, we'll need to amplify the amount of DNA for testing. You should remember that we use polymerase chain reaction, or PCR, for this, but we also want to make sure that we are only working with the DNA from the pathogen and not sequencing the patient's DNA. But how exactly do scientists do this? As you know, PCR produces millions of copies of a DNA sequence in just a few hours. One ingredient used in the PCR process is primers, which are short segments of DNA that are complementary to the template strand to be copied, so when they attach, it provides a double-stranded segment to which DNA polymerase can attach and begin adding nucleotides. Scientists are able to isolate specific target sequences of DNA in this process by choosing primers that specifically attach to segments of DNA that are only present in the DNA of pathogens and not found in humans. For example, all bacteria have ribosomes that are different than any other organisms. Therefore, this part of their genome is unique as well. So if our suspected pathogen is a bacteria, we would choose primers that would bind with the bacterial ribosomal DNA. PCR then proceeds and generates millions of copies of only the pathogen's DNA. Once we have copies of the pathogen's DNA, we can begin the sequencing process. The process called cycle sequencing is similar to a process originally developed by Frederick Sanger in 1977. Once again, we use the process of PCR, but there's a twist. Instead of producing multiple identical copies of the pathogen's entire DNA strand, which we've already done, the goal of a sequencing reaction is to produce a set of DNA fragments that differ in length because each one terminated at a different base. The fragments are then separated by size, and the base at the end is identified, indicating the original sequence of the DNA. Let's look at this process in a little more depth. First, let's zoom into a segment of DNA from the pathogen we just copied using PCR to begin this process. This double-stranded DNA segment is denatured, meaning it's separated into two strands at high temperature. One is called the template strand, and the other is the complementary strand. The template strand is mixed with primers, which we discussed earlier, DNA polymerase, which you'll remember is the enzyme that grabs on and assembles the nucleotides to replicate DNA, free nucleotides, which are also referred to as dNTPs. These, of course, are the bases adenine, thymine, cytosine, and guanine. And here's the twist, fluorescently tagged free nucleotides, which are referred to as ddNTPs. Remember that nucleotides are the monomers that make up a DNA molecule, and they are composed of a phosphate group, a deoxyribose sugar, and one of four types of nitrogenous bases. ddNTPs are similar in structure to other normal nucleotides with two important differences. First, an oxygen is removed from the deoxyribose sugar, and then each type of base has a different color fluorescent tag added so that they glow when run through a laser. For our reaction, the A's will be fluorescent green, the C's blue, the T's red, and the G's yellow. As the PCR reaction runs, the creation of each DNA fragment starts, as usual, with the primer. After the primer attaches to the template strand, the DNA polymerase goes to work and begins to add the complementary nucleotides, creating the new DNA. As you know, whenever there's an A in the template strand, DNA polymerase attaches a T to the new strand. Whenever there's a C, a G is added, and so on. Now, normally in a PCR reaction, this would continue until the entire template strand is replicated. However, because ddNTPs are missing in oxygen, they cannot complete dehydration synthesis to bond with another nucleotide. Therefore, when they are added to a growing DNA strand, the replication process stops and that chain ends. Because there are many more regular nucleotides than fluorescent nucleotides all mixed together, this addition of a fluorescent nucleotide happens randomly at any given location in the chain, which creates fragments of varying lengths. Because millions of strands are created in a PCR procedure, multiple fragments of every length will be created. The result is that each DNA fragment created is a specific length and always ends with a fluorescently tagged nucleotide. When we remove our PCR product from the thermocycler, we have a mixture that contains millions of DNA fragments of all different sizes. So how do we separate different sized DNA fragments? Well, hopefully you're thinking electrophoresis. But instead of using a traditional gel, we use an automated sequencer, which also uses electrophoresis to separate the DNA by size, but it also incorporates a laser, which reads the fluorescent tags at the end of each fragment, and then it sends the results to the computer. Notice the thin capillary tubes inside the sequencer? The tubes are filled with a buffer solution, and the fragments are inserted into these tubes. Let's look at what happens inside one of these tubes. An electric current is applied to the tube so that the negatively charged DNA will move down the tube to the positive end. The shortest segments of the DNA move through the tube the fastest so that they reach the bottom of the tube first. Near the bottom, a laser beam passes through the tube, making the fluorescently tagged nucleotides glow. As they pass through it, optical detectors determine the color of the fluorescence and relay this information to the computer. The color of the fluorescent tag identifies the nucleotide at the end of each fragment. Remember, red is thymine, blue is cytosine, yellow is guanine, and green is adenine. Because yellow can be difficult to see, the computer converts yellow into black. The fragment of DNA that reaches the bottom of the tube first is the smallest fragment. We can determine that its fluorescent tag represents the first nucleotide in a sequence. In this case, the first nucleotide is thymine. Because of its fluorescent tag, it will show up as a red peak. The next fragment to pass by the detector is 2 base pairs long, representing the second nucleotide in the sequence. In this case, our second nucleotide is guanine and will show up as a black peak. As the detector identifies each fragment, we're able to piece together the nucleotide sequence base by base. For our sample, the sequencer generated a sequence of T, G, G, C, A, T, C, A. This is the complementary sequence to our DNA template strand. So this process has allowed us to identify the exact DNA sequence in the sample. Now it's your turn to practice analyzing some sequence data. For the DNA sequence readout shown on the screen, determine the actual DNA sequence. Pause the video and write down the sequence of nucleotides. When you return, you can check your answer. Let's see how you did. The DNA sequence displayed on the screen is CTAGAATGC. Sometimes it's difficult to interpret a nucleotide peak since there are places where the peaks may overlap. This is particularly common at the beginning and the end of a sequence. These ambiguous places are often labeled with an N rather than one of the 4 nucleotides. So now that you know a little bit more about how DNA sequencing works, it's time for you to practice by analyzing the DNA sequence from the pathogen that's making Sue sick. So all the prep work has been done, and you have a PDF that has the data read out from the automatic sequencer. And your job is to write down the exact sequence of the DNA. And once we know what that sequence is, then we can use the tools of bioinformatics to determine the identity of the pathogen. Good luck.